Welcome to the Deep Dive. Today we're going deep into the microscopic world.
Microscopic.
Yeah, of antigens and antibodies.
Right.
We've got excerpts from Microbiology Q&A by OpenStax.
Oh, wow.
And you're going to be amazed by what these tiny interactions can reveal.
I think people are going to be really surprised at just how much is going on.
Yeah.
At such a tiny level.
It's incredible.
Yeah, it's true. The level of precision we're talking about here is mind-blowing.
Totally.
Imagine being able to target a single protein on a virus.
Wow.
That's the power of the techniques we're about to explore.
Yeah.
And by understanding them, we gain incredible insight into how your own immune system works.
Okay, let's unpack this first up.
Yeah.
Polyclonal versus monoclonal antibodies.
Okay.
Our source material describes polyclonal antibodies like a diverse army, with each soldier or antibody targeting different parts of the enemy, which is the antigen.
That's a great analogy.
Isn't it?
Think of it like having a whole battalion deployed to take down a threat.
Oh, wow.
They may not all be the most precise,
Right.
but they cover a lot of ground.
Okay, so kind of like a brute force approach.
Kind of, yeah.
Interesting.
Yeah.
And monoclonal antibodies are like elite snipers, laser focused on the same target.
Exactly.
Okay.
Monoclonal antibodies offer incredible specificity, which minimizes the risk of hitting unintended targets.
So are monoclonals always the better choice then?
Not necessarily.
Okay.
While monoclonals are incredibly powerful, especially when it comes to treating complex diseases like cancer, they can be quite expensive to produce.
Oh, okay.
Polyclonals are often more cost-effective, especially for those initial screening tests.
That makes sense. So it's like a cost-benefit analysis.
Yeah, exactly.
Right.
Sometimes you need the brute force.
Right.
Sometimes you need the precision.
Got it.
Yeah.
Now, how do scientists actually see these antigen-antibody interactions?
That's a great question.
Because we're talking about things on a molecular level here, right?
Right, and that's where the ingenuity of these techniques comes in.
Yeah.
One method is the precipitation ring test.
Okay.
Imagine adding antibodies to a solution with antigens. If they bind, they form a lattice structure.
Okay.
Think of it like microscopic Legos clicking together.
I like that analogy.
And that structure becomes heavy enough to fall out of the solution.
Oh, wow.
Creating a visible ring.
Wait, so scientists can actually see this clumping happen?
They can, and they've even refined this principle with techniques.
Oh, wow.
Like the Out-Ternoni assay and radial immunodiffusion.
Okay.
Where they actually measure the size of these precipitation zones.
Wow.
To quantify exactly how much antigen is present.
So it's not just about detecting the presence of an antigen.
Right.
But also figuring out how much is there that's incredible.
It is pretty amazing.
What other methods are there?
Well, there's another fascinating process called agglutination, where antibodies act like glue, clumping cells or particles together.
So instead of a ring forming,
Yeah.
it's more like a big clump of cells you can actually see.
You got it.
Wow.
Agglutination is used to identify different strains of bacteria.
Okay.
And it's even used to ensure blood compatibility.
Oh, wow.
During transfusions.
That's really important.
The direct Toombs test, which helps facilitate this clumping, is crucial for diagnosing certain diseases where your body might be attacking its own red blood cells.
Okay, I'm starting to see how diverse these techniques are.
Yeah.
We've got precipitation agglutination, all based on the idea of antibodies binding to antigens.
Yeah.
But each with a unique way of making that interaction visible.
Exactly. And it gets even more interesting when we dive into the world of enzyme immunoassays, or EIAs.
EIAs.
These techniques use enzymes, those tiny catalysts that speed up reactions to produce a signal, often a color change, that indicates the presence of a specific antigen or antibody.
Color change. That definitely sounds more exciting than staring through a microscope.
It is.
Tell me more.
Well, one specific type of EIA is called ELISA, or enzyme-linked immunosorbent assay.
ELISA.
In a direct ELISA, you're looking for a specific antigen. The antibody captures it. And then a secondary antibody with an enzyme attached binds to the antigen.
Oh, wow.
This enzyme then acts on a substrate, causing a color change.
So like a chemical reaction you can actually see.
Exactly. The more intense the color, the more antigen is present.
So it's like a color-coded measurement system.
Pretty much.
The darker the color, the more antigen there is.
Precisely. And then you have indirect ELISA, which is used to detect antibodies in a patient's serum.
So kind of the reverse.
In this case, the antigen is fixed. The patient's antibodies bind. And then a secondary antibody with an enzyme detects those antibodies. again resulting in a color change.
So direct ELISAs detect antigens?
Yep.
And indirect ELISAs detect antibodies.
Oh, got it.
You got it. Our source material also mentions immunostaining techniques.
Oh, yes.
What are those?
Immunostaining is a fascinating application of EIAs that allows scientists to visualize cells and structures within tissues.
Oh, wow.
Think of it like highlighting specific parts of a cell under a microscope.
That's so cool.
It is.
Okay, we've covered precipitation agglutination and EIAs. What other techniques do scientists use to see these microscopic interactions?
Well, let's shed some light on the situation.
Okay.
Literally, we can use fluorescent antibodies, which are tagged with special molecules that make them glow under certain conditions.
So instead of a color change, we're talking about glowing antibodies.
Yeah.
Science just keeps getting cooler.
It really does.
So we're literally looking at things that light up under a microscope. That's amazing.
We have direct immunofluorescence, where the fluorescent antibody directly binds to the target.
Yeah.
And indirect immunofluorescence, where a fluorescent antibody binds to another antibody that's already attached to the target.
Oh, wow.
It's like a tag team operation highlighting what we need to see.
Okay, so you can use these antibodies to kind of pinpoint exactly what you're looking for.
Exactly.
In a sample that's incredible.
And they come in so many different colors now, too.
Oh, really?
You can have red and green and blue.
So it's like a rave under the microscope.
It's pretty much a rave.
I love that.
Yeah.
What kind of things can you actually observe with these glowing antibodies?
Oh, all sorts of things.
Yeah.
You can quickly identify bacteria in clinical samples. Detect antibodies in a patient's blood, and even diagnose autoimmune diseases where your immune system mistakenly attacks your own body.
Oh, wow.
It's a remarkably versatile technique.
That is really fascinating.
It is.
Okay, one last question before we move on.
All right.
What about flow cytometry? The source material mentions it as a way to identify and quantify different cell populations.
It's a really powerful tool.
How does that even work?
Imagine a high-tech sorting hat for cells.
Okay.
That's essentially what flow cytometry is.
Interesting.
It's like a cellular census where each cell gets counted and categorized based on its unique characteristics.
Okay, but how do you count and categorize individual cells when they're so tiny?
That's where those fluorescent antibodies come back into play. Cells are tagged with antibodies that bind to specific surface proteins. And then they're passed through a laser beam. The light emitted from the fluorescent antibodies is detected and analyzed, allowing scientists to identify and quantify different cell types within a mixture.
Wow. So you can not only see these microscopic interactions, but you can also analyze entire populations of cells using them.
Exactly. And there's even a more advanced version called a fluorescence-activated cell sorter, or FACS, which can physically separate cells based on their fluorescent properties.
Wow.
Think of it like a microscopic postal service sorting cells into different bins based on their glow-in-the-dark addresses.
Okay, this is officially one of the most amazing deep dives we've done.
I know, it's really cool stuff.
To recap, we've covered an impressive array of techniques.
We have.
Precipitation agglutination EIAs, fluorescent antibodies, and flow cytometry.
It's quite the toolbox.
It is. All of these tools in their own unique way help us visualize and study the interactions between antigens and antibodies.
And what's truly amazing is that these tools aren't just confined to research labs.
Oh, really?
They're being used in countless ways. From diagnosing diseases to developing groundbreaking treatments shaping the landscape of healthcare.
I'm especially curious about the practical applications of all this. How does understanding these microscopic interactions translate into real-world benefits for people?
Well, let's start with diagnostics. These techniques are absolutely invaluable for identifying the specific bacteria or viruses that might be causing an infection.
So like figuring out if someone has the flu or strep throat.
Exactly, but it goes far beyond that.
Okay.
For example, remember those agglutination assays we talked about? Those are used to screen blood for compatibility before transfusions.
Right, to make sure the donor blood won't be rejected by the recipient's immune system.
Exactly.
It's like making sure everyone is speaking the same language immunologically speaking.
Very precisely. And EIAs are widely used to detect the presence of antibodies against specific pathogens.
Oh, wow.
This is essential for diagnosing diseases like HIV or Lyme disease.
So these techniques aren't just for research labs.
Nope.
They're being used in hospitals and clinics every day.
Every day.
To diagnose and treat patients.
Absolutely. And their impact goes even deeper in the realm of disease treatment.
Okay.
Monoclonal antibodies in particular are changing the game when it comes to how we approach certain conditions.
Okay, let's dive into that. We touched on monoclonal antibodies earlier.
We did.
But I'm really eager to learn more about how they're being used therapeutically.
Well, imagine this.
Okay.
You create an antibody that specifically targets a cancer cell.
Wow.
Leaving healthy cells completely unharmed.
Oh, wow.
That's the power of monoclonal antibody therapy.
So it's like a guided missile that seeks out and destroys the bad guys.
That's a great analogy.
That's amazing.
These monoclonal antibodies can be engineered to deliver toxins directly to cancer cells.
Oh, wow.
Or they can act as a flag, marking those cells for destruction by the immune system.
So it's like a double whammy.
It is.
Wow, that sounds like a much more targeted approach compared to traditional chemotherapy.
Right.
Which often comes with a lot of side effects.
Exactly. And monoclonal antibodies are also being explored as treatments for autoimmune diseases.
Okay.
Where your immune system is mistakenly attacking your own body's tissues.
So instead of killing cancer cells, they could be used to calm down an overactive immune response.
Precisely, they can block the signals that trigger inflammation.
Okay.
Or target specific immune cells that are involved in the autoimmune attack.
This is all truly groundbreaking.
It's an exciting field.
Our source material did mention that monoclonal antibodies can be expensive to produce, though.
Yeah, that's true.
Does that limit their use?
Cost is definitely a factor.
Yeah.
But the potential benefits are so significant that research is constantly pushing towards more cost-effective production methods.
It's encouraging.
It is.
So from diagnosing infections to treating cancer and autoimmune diseases, the applications of these techniques are vast.
And we haven't even scratched the surface. These tools are also instrumental in basic research, helping us understand the intricate workings of the immune system on a molecular level.
It's amazing to think that something so small can have such a profound impact on our understanding of health and disease.
It really speaks to the ingenuity of scientists, the incredible power of the immune system itself.
Absolutely.
You know, our source material, Microbiology Q&A, actually includes some review questions that I think could really help solidify your understanding of these concepts.
Okay.
Would you be interested in tackling a few of those?
Absolutely. I'm feeling like a microbiology detective ready to crack the case.
Great. Let's start with a question about the specificity of those polyclonal antibodies we talked about earlier.
Ooh, I remember we discussed that polyclonal antibodies can sometimes bind to unintended targets, right?
Yes, exactly. That's what we call cross-reactivity.
Okay.
So thinking back to our analogy, it's like one of those battalion soldiers accidentally hitting a friendly target.
Okay, that makes sense. What's the next question?
This one asks how monoclonal antibodies are produced.
Okay.
Remember that fascinating process of fusing antibody-producing cells with myeloma cells to create hybridomas?
Yes, those hybridomas are like tiny antibody factories churning out identical copies of a specific antibody.
Precisely. It's a truly remarkable feat of bioengineering. Ready for another one?
Bring it on.
All right. This question mentions the VDRL test. Do you remember what that's used for?
That one rings a bell. It's a test for syphilis, right?
You're exactly right.
Okay.
And it relies on the formation of flocculants.
Okay.
Those visible clumps of antigen antibody complexes that we talked about earlier.
Yeah.
It's another example of how we can make those microscopic interactions visible.
Okay. I'm starting to get the hang of this. Let's see what else we've got. All right. Hit me with another question.
Okay. This one dives into the Outerloni assay.
Okay.
Where we see that sharp precipitin arc forming between the antigen and the antiserum.
Yeah.
The question is, why does that arc stay visible for so long?
I want to take a stab at this one.
Okay.
It's because those antigen-antibody complexes are forming that lattice structure, right? That microscopic net we talked about.
Yeah.
Is it because it's too large to diffuse through the gel?
You nailed it.
Really?
That precipitin lattice is just too big to move easily through the agar gel. So that arc remains visible.
So it's like a microscopic fingerprint left behind by those antigen-antibody interactions.
Exactly.
That's so cool.
It is pretty neat.
This is making me feel like I could ace a microbiology exam.
I think you could.
Okay, what's next?
This next question shifts gears to bacterial identification.
Okay.
It says, we use antisera to distinguish between different within a species of bacteria.
Let me think.
Take your time.
Different strains, like even within the same species of bacteria, there can be slight variations.
Exactly. We call those variations serovars.
Serovars.
They're strains of bacteria that differ in their surface antigens.
Okay.
And we can use specific antibodies to identify them.
So, like, even though they're the same species, they have these little differences.
Right.
That make them unique.
Exactly. It's like having a key that only fits a specific lock.
Oh, that makes sense.
Yeah.
Okay, I'm ready for the next challenge.
Building on that idea of bacterial identification, this question asks what we often link antibodies to. In order to better visualize agglutination.
Okay.
Remember agglutination is that clumping we talked about?
Yeah.
We need something that would make those clumps easier to see, right?
Right.
What about something like latex beads? Would those work?
You are on a roll. We often link antibodies to latex beads.
Yes.
These beads provide a larger surface area, making the clumping more visible to the naked eye.
It's like attaching tiny flags to those bacteria so we can see them waving around.
I love these analogies. They really help me visualize what's going on.
Good. I'm glad.
Okay, I think I'm really starting to understand how these techniques work.
That's great.
What else do we have?
Let's dive into blood typing with this next question.
Okay.
It focuses on the antibody screening test that's done before transfusions.
Oh, right. That test makes sure that the recipient isn't making antibodies against any antigens outside of the ABO or RH systems, right?
Exactly.
Kind of like a double check to make sure the blood is compatible.
It's an added layer of safety to prevent complications during a transfusion.
That's really important. What's the next question about?
This one asks about the purpose of the direct Coombs test. Do you remember what that one is used for?
If I recall correctly, that test helps determine if someone is producing antibodies that attack their own red blood cells.
You're exactly right. It's a key diagnostic tool for certain conditions where the immune system is mistakenly attacking the body's own cells.
Wow. So we can actually use these tests to figure out if someone has an autoimmune disease.
We can.
That's incredible. Okay, moving on. What's next on our microbiology quiz show?
This one focuses on viral hemagglutination assays.
Okay.
It states that these assays only work with certain types of viruses.
Okay.
Why would that be?
That's a good one. I think maybe it has something to do with the virus's ability to bind to red blood cells.
You're on the right track. These assays only work if the virus can directly cross-link red blood cells, causing them to clump together.
Okay.
It's like the virus needs a specific hook to grab onto those red blood cells.
So it's all about those specific interactions between the virus and the cells.
Exactly. Each of these techniques relies on understanding the very specific ways that antigens and antibodies interact.
I'm starting to see a theme here.
It all comes back to those interactions.
This has been an incredible journey through the world of antigens, antibodies, and all the amazing techniques used to study them.
It has been a deep dive.
It really has. Who knew the microscopic world could be so fascinating?
It really is remarkable. And what's even more impressive is how these tools, which started as scientific curiosities, have become absolutely essential for diagnosing and treating diseases, truly shaping the future of healthcare.
It just goes to show you the power of scientific exploration. When we dare to delve deeper into the mysteries of the natural world, who knows what incredible discoveries await us.
I completely agree. And that's the essence of the deep dives, sparking curiosity, deepening your understanding, and leaving you with a sense of wonder about the intricate workings of the world around us.
I have to say, this deep dive has certainly left me with a lot to ponder. What stood out to you most from our exploration today?
For me, it's the sheer elegance and complexity of the immune system.
Yeah.
These tiny interactions happening on a microscopic scale are orchestrating this incredibly intricate defense system that keeps us healthy.
Wow.
It really is a testament to the power of nature.
I couldn't agree more. Until next time, keep diving deeper